मॉर्निंग टू ऑल. सायन्स फोरम नाशिक आणि LG मोबाईल लॅबोरेटरी, सावित्रीबाई फुले पुणे विद्यापीठ यांच्या संयुक्त विद्यमाने आपण हा ऑनलाईन छंद वर्ग 3 मे पासून सुरू केलेला आहे. या आजच्या कार्यक्रमासाठी आजचे आपले तज्ज्ञ मार्गदर्शक प्राध्यापक श्याम आचार्य सर, तसेच सायन्स फोरमचे सदस्य, इतर विज्ञानप्रेमी, शिक्षक, विद्यार्थी, मी आपणा सर्वांचं स्वागत करतो. विज्ञान गंगा ऑनलाईन छंद वर्ग हा 3 मे रविवारी 3 मे पासून आपण सुरू केलेला आहे. पहिलं पुष्प डॉक्टर जयंत जोशी भाभा ॲटॉमिक रिसर्च सेंटर यांनी सादर केलं होतं आणि आजचं दुसरं पुष्प सादर करण्यासाठी आपल्याकडं प्राध्यापक आचार्य सर उपस्थित झालेले आहेत. आजच्या या कार्यक्रमासाठी 5,000 विद्यार्थ्यांनी त्याची नोंदणी केलेली आहे. हा उपक्रम यूट्यूबवरती लाईव्ह दिसणार आहे. या कार्यक्रमाचं साधारण स्वरूप म्हणजे साधारण 45 ते 50 मिनिटं किंवा 1 तासापर्यंत लेक्चर किंवा मोस्टली डेमॉन्स्ट्रेशन असणार आहे. विद्यार्थ्यांना जर का काही प्रश्न असतील तर ते त्यांचे प्रश्न चॅटबॉक्समध्ये टाकू शकतात. ते आमच्यापर्यंत पोहोचतील आणि कार्यक्रम झाल्यानंतर, डेमॉन्स्ट्रेशन झाल्यानंतर त्या प्रश्नाची उत्तरं ही दिली जातील. आजच्या या कार्यक्रमासाठी विज्ञानप्रेमी, बायोलॉजी विषयाचे तज्ज्ञ माननीय प्राध्यापक श्याम आचार्य सर आपल्याला मार्गदर्शक म्हणून लाभलेले आहेत. बायोलॉजी, लाईफ सायन्स या विषयाचे ते तज्ज्ञ आहेत आणि हा विषय शिकवण्याचा अत्यंत दीर्घ अनुभव त्यांच्याकडे आहे. त्यांचे लेक्चर्स खूप इंटरेस्टिंग आणि ज्ञान प्रबोधक होत असतात. कुठलाही अवघड विषय सहजतेने उकलून तो सोपा करून सांगण्याचं कौशल्य हे त्यांच्याकडे आहे आणि हे आपल्याला या आजच्या सत्रातून आपल्याला त्याचा अनुभव येणारच आहे. आजच्या या कार्यक्रमामध्ये आपण हा जो उपक्रम घेत आहोत त्याच्यामध्ये 2 ग्रुप आहेत. पहिला ग्रुप साधारण 1ली ते 5वी 6वीच्या विद्यार्थ्यांसाठी आहे आणि दुसरा ग्रुप हा 6वीच्या पुढील विद्यार्थ्यांसाठी आहे. आजचा हा कार्यक्रम विशेष करून 6वी 7वीच्या पुढील विद्यार्थ्यांसाठी आहे. त्यांना तो जास्त चांगल्यापैकी समजेल आणि त्यांच्या सिलेबसच्या रिलेटेड, जनरल नॉलेजच्या रिलेटेड हा कार्यक्रम असणार आहे. त्यामुळे हा आजचा जो डेमॉन्स्ट्रेशनचा प्रोग्राम आहे हा खूप इंटरेस्टिंग आणि खूप उपयुक्त असणार आहे. तर हा आपण कार्यक्रम आजचा सुरू करत आहोत. तर मी प्राध्यापक डॉक्टर प्रवीण जोशी यांना विनंती करतो की त्यांनी आपले जे विषय तज्ज्ञ आहेत माननीय आचार्य सर यांची थोडक्यात ओळख करून द्यावी आणि म्हणजे आपण हा कार्यक्रम पुढे सुरू करू.
प्राध्यापक श्याम आचार्य सर यांची थोडक्यात ओळख करून देतो. तर MSc झूलॉजी, फर्स्ट मेरिट इन द युनिव्हर्सिटी डिपार्टमेंट. त्यानंतर सरांचं MAd इन एज्युकेशनल सायकॉलॉजी अँड स्टॅटिस्टिक्स, त्यामध्ये पण सर सेकंड मेरिट इन द युनिव्हर्सिटी. सर MPhil इन एन्व्हायरमेंटल सायन्स ग्रेड A मध्ये झालेले आहेत. बायोलॉजी टीचिंग लर्निंग सरांचा एक्सपिरियन्स जवळजवळ 53 वर्षांचा आहे, 1973 टिल पुढे. त्यामध्ये 1973 1973 to 1976 सर वॉज लेक्चरर ॲट नागपूर. देन 1976 to 1978 lecture at Assam. Then 1978 to 2004 lecturer in HPT Arts and RYK Science College and SMRK College at Nashik. Then from 2004 to till today independently established and conducted biology based science activities, biology learning center at Nashik, study travels. त्यामध्ये visited to iconic places of science research in the past such as Cambridge, Oxford, Harvard, UCLA, Stanford and many national research institutes within India. Also visited for study purpose mainland, original workplace at Czech Republic, then Darwin original research place near London, Marie Curie Museum and her original research place in Poland. तर या प्रकारे सरांचं गेल्या 53 50 वर्षापासून सर टीचिंग फील्डमध्ये बऱ्याच विद्यार्थ्यांना त्यांनी ट्रेन केलेलं आहे. बरेच विद्यार्थी अगदी मोठ्या पदावर गेलेले आहेत. सर सायन्स फोरमचे पहिल्यापासूनचे मेंबर्स आहेत आणि आजचं तुम्हाला लेक्चर सरांचं रिलेटेड टू ब्रेन हे नक्कीच आवडेल.
धन्यवाद सर. आजच्या या सत्राला आपण सुरुवात करत आहोत. नो युअर ब्रेन या विषयावरती मार्गदर्शन करण्यासाठी मी आचार्य सरांना विनंती करतो.
The goat brain, oh my god, that's something. So here we have a goat brain and we are going to take into account the various aspects of the goat brain. So this is going to be anatomy of goat brain basically. Even though it's a primary and a foundation course, and because you have learnt and heard of these parts of the brain for several days in your schools and books, I think it will be good if you have an opportunity to look at the brain first time, maybe most of you. Okay, let's now understand. This is the picture of the goat brain, and this will very much match if I allow you to look at this particular way. Look at this. So you have now the cerebral hemispheres, and these are the cerebral hemispheres, and you have the cerebellum here, and you have the cerebellum here, and lots of other details which I can show closely. So this has been taken up from the net. This is goat brain, the sheep brain actually, but sheep and goat brains will be almost very much identical. So I would now go into some details of the brain. You already know that this is something which is present in the cranium, that is a brain box, which is a part of the skull. Here now I would like to point out some of the parts. Here you have the cerebral hemispheres, the two cerebral hemispheres. The two cerebral hemispheres, which are the main aspects of the brain that you know very well. You have the cerebellum. This is the cerebellum. So cerebral hemispheres and cerebellum. Cerebral hemispheres are folded. You know the sulci and gyri. Do you know these terms? Sulcus and a gyrus. So these are the sulci and gyri, and that is to increase the surface area. And why to increase the surface area? If you have listened carefully to the lecture, it was mentioned that most of the cell bodies of the neurons are seated onto the surface. So billions of cell bodies are located on the surface, and their axons are deeper down. And to accommodate so many cell bodies, you need to have more surface area, but the overall size of the brain cannot be increased simply because there is a limit to the size of the head and so size of the brain. And so naturally the only option is to have more surface area, you must have folds. So these are the folds. Even the cerebellum will have folds. I will now simply say that here, if you can observe, this is the midline, and this is what is called as medial longitudinal fissure. This one, you can observe it here. This is the medial longitudinal fissure, this one, which divides the two parts of the cerebrum, each part being known as cerebral hemisphere. Now, each cerebral hemisphere has lobes. Do you remember? It was told to you. And whatever we learned for the human brain is applicable for the goat brain. At least the morphological details, the anatomical details. And whatever is applicable for the goat brain is applicable therefore for the human brain, general idea, general structure. Here you have the frontal lobes. Then you have the parietal lobes. frontal lobe, parietal lobe. This is the occipital lobe. Frontal, parietal, occipital. And side lobe is temporal. Let's have it from this side. Once again, frontal, parietal, occipital. Frontal lobe, parietal lobe, occipital lobe, and here is the temporal lobe. So these are the lobations. And here deep inside, there is a structure which is known as diencephalon, which is not seen from here, but I'm just pointing out the location. Deep inside here, there is diencephalon. Actually, if I hold it this way, you can now have a picture. This is the ventral aspect of the brain. So you have now here the ventral aspect of the brain. So you will find once again I'll point out to the temporal lobe. You can see from the side the temporal lobes are there. Okay, and lower side of the frontal lobes is there. As you can see here, what are these two? Look here. These are the olfactory tracts. These are the olfactory tracts, and then the olfactory nerves are cut here. This is a mammillary body. Can you see this double structure here? This is a mammillary body. And although because the pituitary usually remains within the skull when the brain is taken out, so otherwise I'm just showing you the position. Here is a position where the pituitary gland was present. Then you can imagine this is the pons. This is the pons varoli, pons and the medulla oblongata. Pons, medulla oblongata, and part of the cerebellum. Once again, this is the ventral view, or you can say the inferior view of the brain. And this is the superior view, that is the dorsal view of the brain. There can be many details, and we are not going to take any section here. I will not go into further details. I think it is possible that we will keep this information in mind and then go for observation of the human brain. But I'm sure you are not expecting actual human brain, because that's not possible. That's not possible. It's not possible to have a human brain here. So goat brain you have seen. For human brain, I think mostly we'll see the pictures with their small paws. Let's have now some revision about the human brain, by way of pictures, of course, mostly. But then we will still have the goat brain just as a side reference. You're all aware that human brain is located in brain box. As you can see in this picture, this is the skull, and part of the skull is the brain box. So all this is the part of the skull in which brain is located. It's called as cranium. Cranium, that is brain box. And then from the brain, the spinal cord begins, and the spinal cord is present in vertebral column, which is called a spine. So it's very well protected. I think it will be good if we once again have a look at the skull. I'll show you the skull. So here is the human skull, you are aware, and the cranium is the brain box here. I'll just open the cranium, and it's empty now. It's empty because the brain has already been taken away long back when the skeleton was being made. So once again I say that this comes from the medical college, and this was obtained by Dr. Satyajit Joshi, a famous orthopedic surgeon of Nasik, and who has been my friend, and once upon a time he was my student, I told you already, and we always meet together, and we remember those days when we started using this long back in 1997-98. So this particular skeleton has been used by thousands of students. They have actually handled. You want to handle them? You can always handle them. So this is the skull and this is the brain box. See, skull has many bones. There are facial bones. These are all bones of the face, facial bones, and then the cranial bones. I'll just make a mention of the few bones of the cranial box. You know, these are bones have the same names as the brain parts. So these are the two frontal bones, these are the two parietal bones. Once again, the frontal, the parietal bones, and then you have the occipital bones. So frontal, parietal, and occipital bones. Similar names are there. You know, when you think of the brain parts, you have frontal cerebrum parts, frontal lobe, parietal lobe, occipital lobe. So frontal bones, parietal bones, and you have the occipital bones. So names of the bones and the lobes of the cerebrum, the names match, of course. When you think of the floor, this is a floor of the cranial cavity, and this, can you see this complete Y shape? The bone which is like a butterfly. This entire butterfly-like bone, this whole thing, butterfly-like bone is being called as sphenoid bone. What's the name? Sphenoid. And in front of this sphenoid bone, there is one more bone, and that is called as ethmoid. Sphenoid and ethmoid. And there are other bones, don't worry about it. What is important for us to understand, that there is a cavity in the sphenoid bone. Can you see this cavity? This tiny cavity? It was shown to you during the lecture. Do you know what is this called as, by chance, if you know? Yes, some of you have said, sella turcica. Okay, so it is here that the pituitary gland is located. You can imagine the brain is here. Imagine this brain is here. Oh my God, I can't put a goat brain into human skull. That's not possible, and that's not desirable. But you can imagine the pituitary gland will be on the underside of the brain, and human brain will be much, much larger, of course, and this is how it will be present. And the pituitary gland will be lodged, it will be placed here in this cavity, the sella turcica. That is the reason when brains are taken out, if enough care is not taken, then the pituitary gland remains there because it gets cut off. And that's how here, in the case of the goat brain, pituitary gland is not seen. So this is the thing. And can you see this big gateway? You know, it is this gateway through the spinal cord comes out. So you can say that spinal cord will come out of this one. This is the gateway through which the spinal cord will come out. This one. So the brain is located here, the spinal cord then will pass out here. So once again, imagine this is the brain box, and it will be the brain located here, and you can see here this passage through which the spinal cord will come out, and immediately there is a vertebral column out there. So that is about the brain box, and then we can have some of the pictures. Look at this particular picture. Here you can observe very clearly the Celsia and the gyri. You know, a slice has been taken up here. Frontal lobe, parietal lobe, the greenish occipital lobe. Symbolic colors, of course. And a slice has been taken out here to indicate what is a sulcus and a gyrus. This elevated portion, the fold, is called as gyrus. And between two gyri, gyrus is singular, gyri is plural. Between two gyri, there is a sulcus. So sulcus is depression and gyrus is an elevation. So the entire brain is folded, and there are lots of interesting things that you might have heard about the folds. That the more the folds, more the intelligence. And Einstein's brain, this and that. Well, I'm not going into those details, but there is some limited, there's a limited importance to those things. Cortex and medulla. Look here, it was told you that a few millimeter cortex is there, and then inside there is a medulla. Well, cortex is that where the cell bodies are present. It was clearly told to you in the lecture that cell bodies are present here, millions and millions, billions actually. Cell bodies will be present here, and fibers will run inwards. There are many details of this anatomy of the brain, and we are doing it very much superficially. Longitudinal fissure, this one, and it was already told to you, even in the case of the goat brain, there is a longitudinal fissure here, there is a longitudinal fissure here. Okay. We will now have one more picture. You are all aware that mapping has already been done for human brain, and this is something where you can find out the different areas. Basically, first let us find out, this is the front side, anterior side. So this is frontal lobe, and then this is the parietal lobe, and this is then the occipital lobe. So frontal, parietal and occipital lobes are there. You can see the parietal lobe here. So frontal, parietal and occipital lobes, and you have some centers. For example, there is what is called as motor speech area. If I'm talking just now, it is because my motor speech area, which is called as Broca's area, is sending signals to my speaking apparatus, that is larynx, and also to my tongue, and also to my muscles of the cheek and related other muscles which make possible the speech. So all my signals for the speech which I am doing just now are coming from Broca's area. It's a motor area because it is sending motor signals, the orders. Then you have association area, which is located here. That is called as auditory association area because there is an auditory area here. So hearing is possible here. Then you have visual area. Visual area is that which is on the back side of the occipital lobe, and the visual area is meant for ability to perceive vision. You know, the perception is something where you experience that particular sense. Although eyes are getting the photo stimulus, light stimulus, and the eyes are able to convert that photo stimulus into electric current, and electric current has, imagine it's already reached the brain. It is here that it will be integrated, perceived, and so this is the area which is called as the vision area. So lots of mapping has been done. So it was not easy, you know, all these various areas, 44 areas have been indicated here, and all these areas have been mapped over the last more than 100 years, and that has been done by animal experimentation, putting electrodes, rarely on humans also indirect data can be obtained, and also understanding what way humans have these areas, particularly some of the patients who suffer accidents, and then what particular brain part is damaged and what function is lost. There are lots of complicated things. I think we can have now the underside, the lower side of the brain. We have already seen this for the goat, and now you can have a clear picture. Here you have now once again the cerebrum part, and then you have the cerebellum part. This is cerebrum and this is cerebellum, and then you have the pons, and then you have the medulla oblongata, and then you have the spinal cord which begins here. Okay, once again, the big thing, major part of the brain is cerebrum. This is from the underside. Cerebrum, then cerebellum, then this is pons, and then this is medulla oblongata. That's the last part, and from where the spinal cord begins. So I think it is possible to once again consider the scene, which is rather similar, although this is a picture from the Tortora's book, Human Brain Underside, and this is the goat brain, but I can point out a few similarities. So this is the pons here, and here is the pons. Unfortunately, this has been much damaged now, so things are not very clear. I would like to point out to these nerves. You already have learned that from the brain there are 12 pairs of nerves starting, and here are those 12 pairs. I'll just read out their names. You don't have to worry, you don't have to remember just now, but during your regular course, of course, you will be told more about them, and you will be then remembering them in sequence. If you're interested, I can mention those names and you can repeat after me. First, I'll read all of them. 12 pairs of cranial nerves is peculiarity, and this is the first nerve, olfactory. And as the name indicates, it is something which is getting the stimulus, the sensory stimulus from the nasal capsule by way of olfactory bulbs. And then the olfactory nerves are there, and olfactory nerves will carry the message to the brain. Then you have the optic nerve. These two are optic nerve, and they make a cross. That is called as optic chiasma. Don't worry, you don't need to remember everything, but some of these pictures are there in your books also. Not this one, maybe the lateral view or the front view or maybe the top view. So once again, olfactory, then optic, then oculomotor, this one. This is a nerve which is supplied to the eye for the movement. Also then, you have the trochlear, this one. This trochlear is again a thin nerve which also supplied to the muscles of the eye. Then comes one of the thickest nerve, trigeminal. And this is the nerve, trigeminal, which is supplied to the jaws and part of the tongue also. And this is very important nerve from medical point of view, dentist point of view, because it is this nerve which has supplied to the with the gums also, and then the anesthesia which the doctors give will make this supply of this nerve numb. Then abducens, one of the shortest nerve is abducens, then facial, and then vestibulocochlear, the eighth nerve, that is actually concerned with hearing, cochlear, cochlea, and vestibular is related to the balancing. I think you will definitely, hopefully, you will have observed the lecture, the second part of the nervous coordination lecture, and that time it was told to you that from semicircular canals there is a nerve coming up, and also from the cochlea there is another piece of the nerve coming up, and the two meet together and they run to the brain. So there is a vestibulocochlear nerve which is 8th nerve. Glossopharyngeal, the 9th nerve, the glossopharyngeal. And then you have the vagus nerve, the 10th nerve. I put a star mark here because vagus nerve is that which goes to the inner organs. Branches of the vagus nerve, they go to the stomach, intestine, liver, pancreas, lung, heart. Vagus nerve is important. It's the longest of the cranial nerves. And there are others like accessory, spiral accessory and hypoglossal. You don't have to remember all of them, but I'll just utter once, and if you are interested, you can repeat after me. All of you say olfactory, optic, oculomotor, trochlear, trigeminal, abducens, facial, vestibulocochlear, glossopharyngeal, vagus, accessory, hypoglossal. One thing, mammals have 12 nerves, birds also have the 12 nerves, the same sequence, and reptiles also have 12 nerves and the same sequence. And most of the parts of the brain of the reptiles, birds and mammals are very similar. And this is not something that you are hearing for the first time. What you will call all this similarity? Yes, it will be called as homology. So basically, just like heart structures are homologous, the brain structures are homologous, the nerves are homologous. The number of nerves, the cranial nerves, which are starting from the brain, their number is little less in fishes and amphibia. Fishes have only 10 pairs of cranial nerves, and amphibia also have 10 pairs. But reptile, birds and mammals have 12 pairs. So that's about the arrangement. I think I'll just take this opportunity to show you a little complicated thing. You may probably wonder what it is all about, but interesting thing is that attached to the lower side of the brain here, there is the pituitary gland, and you know this very well. And there is a stalk here, the infundibulum, and to which the pituitary gland is attached. All this is hypothalamus. I think this hypothalamus is very famous, and you already know. And in the worksheet, a question has been raised about the hypothalamus. You know, hypothalamus is a region which actually originally belongs to nervous tissue. It is a part of the brain. But hypothalamus also secretes some hormones. In the hormonal coordination lecture, it was told to you that some of the hormones of the hypothalamus, they control pituitary. So this is a part, it originally belongs to brain, that is nervous system, but because it also produces some hormones which control the pituitary, you can say that this also belongs to endocrine system. So this is something which is common between the nervous system and endocrine system. I am talking of hypothalamus. If this is hypothalamus, then this is hypophysis. You know, pituitary gland is called as hypophysis. And this is the optic nerve. And there are lots of things which I don't want to repeat them, but pineal gland is something you may be interested in knowing, because you know pineal gland is something which makes our rhythm, you know, the circadian rhythm, the 24-hour rhythm. And in the lecture it was told to you that it is the pineal gland which produces a hormone known as melatonin, and it is required for inducing sleep, and there are lots of other functions, of course. Quite complicated, you know. In your first year MBBS itself, you will have lots of details about the brain that you are going to learn. And yes, these things which you are learning will be helpful to you. Even if you don't want to go to medical, that doesn't matter, because knowing about brain is one of the wonderful opportunities. I have one more picture to be shown to you, and that's about limbic system. So here it is. Limbic system. In the lecture, you might have heard that there is something like limbic system in the brain. I'll just make a comment about the limbic system. Basically, all the green things which are shown here, they are actually part of the cerebrum, and part of the other regions. And I think you will have some interesting names heard. Amygdala. And then hippocampus, this one, hippocampus, and such things. There are lots of areas here, and all these areas taken together, they make what is called a limbic system. Actually, these are parts of the brain, otherwise also doing several functions, but it appears that the limbic system, which is now shown here by these this green shading, is something which is, so to say, called as emotional brain. And that PDF sheet is with you, and you will read that few lines about this emotional brain. Why it is called as emotional brain? Because it is something which controls your behavior. It is something where primitive instincts like fear, rage, aggression, to some extent memory, and to some extent learning is also possible. This is a very important part of the brain, because that saves life, you know. It is a limbic system which helps us to ultimately defend ourselves. If you have seen a cat, if the cat is not friendly with you, and if you try to harm the cat, then it will probably try to behave in such a way that you will be afraid a little bit, suppose, because if you are a new person to that cat. So that is all, you know, the animal instinct. They just want to save themselves, and they want to behave in a way which will be probably either an offense or a defense, and so on. So limbic system is very important. It's emotional brain, and that's an important part, but it is a primitive thing. That's very important. I think with this, with a pause, I can go for a few more pictures. A little bit of understanding about the development of the brain. I must mention very specially here that brain is the first organ which starts developing. That's very important because it has been shown that the developing brain will also be able to regulate the developing body, and so heart and brain they start functioning quite early. The entire nervous system is formed from what is called as ectoderm. Do you know by chance this term? Ectoderm, mesoderm, endoderm. If you know, good. If you don't know, no problem. But when the fertilization occurs and the zygote starts developing and a very tiny embryo is produced, there are 3 layers of the body of the embryo: outer ectoderm, middle mesoderm, and inner is the endoderm. So don't worry about it. But the entire nervous system is formed from ectoderm. So this is how it begins. This is the beginning part of the brain, which is called as forebrain. This is the middle part, which is called as the midbrain. And so the midbrain and the hindbrain. So forebrain, midbrain, hindbrain. These are the 3 parts which can be recognized quite early. This is less than 1 month. This is a 3 to 4 week old embryo. And this is a 7 week old, less than 2 months. And see how fast this develops. This is the forebrain of the first month, and this is the forebrain of the second month. You can see the colors will tell you the midbrain and then the hindbrain. The brain is growing very fast. Now, consider this is a situation which is less than 3 months, and you already have the forebrain develop much more, and the midbrain and the hindbrain. And the baby is now taking a proper shape, and brain is very important. It is during this time that there must be proper nutrition for the mother, there must be proper thyroid function, proper iodine supply, then thyroid will work, and thyroid is required for the development of all body parts and definitely for the brain. Then if this is less than 1 month, this is little less than 2 months and little less than 3 months, and then 9 months over, and at the time of birth the brain is ready. So imagine forebrain develops this much, and all this is forebrain. You can see very clearly here the cerebrum, and you can also like to look at the cerebrum and cerebellum, and you have the cerebrum here and the cerebellum here. All parts are well developed, and the medulla oblongata and the spinal cord. It's very interesting. If the developmental studies of the brain are done for different vertebrates, again, surprisingly, there will be a lot of similarity. Please understand. So the way the human brain develops is all the same, the way the brain develops in the other animals. Yeah, sometimes we do have, we bring the fertilized eggs of the birds and try to allow them to grow here, and then we are able to take out the embryo and study those things. Actually, we really are missing you people here, to be very frank. But then what to do? So we have to continue with this kind of demos, and even this is good. One good thing for you is that when there are 20-25 students at a time, because we have lots of batches for that, then not everybody is able to look at those things with comfort. Now, being this particular situation of online, each one of you has got this opportunity as if the whole thing is going on only for you, so you can have close looks. So that's a benefit. So that's a great advantage of online. But then, of course, the shortage is that you cannot be here. I think, and we really want, because we are having a lot of enthusiasm to arrange these things. And maybe that when you come in March, April for 12th, that time maybe that some of these things can be repeated. Even your senior batch, who is now undergoing their 12th online, they have done many of these things, but then this time they have missed the dissection of the bird. because it was planned in the month of April, and that bird dissection, of course, they have missed. But for them, I don't know when we are going to arrange it. Maybe possible later on. So I think these are very important things for AV science. The science is learned through actual experiences of this kind, and maybe that once you join in for the 12 sessions, maybe some of the 11 things we can repeat, but I'm not sure. I mean, nothing can be guaranteed just now, but we would definitely love to do all that for you. So this is something, and now I'll go for a very different aspect. Here I have now about the brain waves, and you can understand this is what is called as EEG. You know, ECG is different. ECG is electrocardiogram. This is EEG, electroencephalogram. Encephalus, you know, the head. It is possible that electrodes are placed on the different parts of the head, and then electrical activity of the brain is recorded. Wonderful. Heart has electrical activity, brain also has electrical activity. Electrical activity of the heart can be recorded, and that record appears as electrocardiogram. And the electrical activity of the brain can also be recorded, and that is called as EEG. Yeah, it has got a lot of uses. 4 patterns are shown here: the gamma waves, the beta waves, the alpha waves, theta waves, and delta. I'll repeat: gamma, beta, alpha, theta, delta. I know some names are given. If you're learning just now, and I'm trying to say something. So if electrodes are attached to my brain, I mean my head, you will get these kind of waves. If electrodes are attached to your head and map is taken, you'll also get this kind of picture where, you know, it is a very high level brain activity. Then suppose you are just chatting with each other lightly. Even then your brain is busy, but not as busy as it is in the process of learning. So this may be busy and you are talking with someone. So these are beta waves, and the frequencies are mentioned here, which I am not going to talk. When you are relaxed, suppose you are doing meditation, then your waves will become slow, and their amplitudes will also change, and these are the alpha waves. And sometimes, you know, you start feeling sleepy, and then if that time the waves are recorded, then these are the theta waves, which look like this. And when you are fast asleep, then these will be the delta waves. Wonderful conclusions can be drawn by recording the waves, and certain diagnosis of certain disorders can be done with the help of the brain waves. I think here we can have a break, stop, and then continue. Would you like to look at the brain of some bird? Okay, let's have it. Interesting. So here I have a sample of the brain of the bird. This is it. Look here. I'll show it this way to you. And this is the beak of the bird. So it's towards me. And you can see we have cut open the skull. And here you can observe these 2 big things. What are they? Imagine cerebral hemispheres. 2 cerebral hemispheres, and this is cerebellum. Cerebral hemispheres and cerebellum. Other parts are not being exposed. I'll just keep for your reference this brain of the goat side by side. So I'll keep for a big comparison the goat brain along with the brain of the bird. And you can compare. Oh my God! This is a hen, and so bird is a hen here. Cerebral hemispheres, these 2, and these are the cerebral hemispheres, these 2. And this is the cerebellum, and this is the cerebellum. Homology, perfect homology. And human brain actual sample we don't have, but then you can also expect that arrangements will be similar. Cerebral hemispheres too. These are the two cerebral hemispheres. Cerebellum, this is the cerebellum. Birds also have very nicely developed optic lobes, which we are not able to show them here. But because usually birds depend more on the sense of sight. So that's about the bird brain, and with a pause we'll continue. So let's revise a few things about the eye. I have decided to go into some details of the eye, the model, and then I can show you the actual eye of the goat. So you can understand the outermost is the sclerotic, the sclera, and the front part of the sclera is this one, which is transparent and is called as cornea. So sclera, and the front transparent part of the sclera is cornea, and this middle layer, which is shown here with pink, actually it is black. Here in the model it is being shown with pink, and this is what is called as choroid. So sclera, then choroid, and here this yellow thing is all retina, and retina is very nicely supplied with blood vessels. Actually these belong to the choroid layer. So this is the arrangement, and there is a lens here. You can see this lens. So this lens is present here, and around the lens, as you can see here, the dark brown thing, that's iris diaphragm, and the hole through which the light enters, that hole which you can see here, this hole I'm talking about, what you will call this as? This hole you will call it as pupil, through which the light enters. I'll put back the lens like this. Optic nerve. You can see here the optic nerve, which is emerging out of the eye. The retina has rods and cones, which are not shown here, and there are several other layers of the cells, but important cell layer is rods and cones. And from the outermost layer of the retina, the neurons begin, and the axons they make an exit, and is the optic nerve. I'm going to show you this optic nerve in the goat's eye. So this is the generalized model, and there are muscles, and these muscles are supplied by cranial nerves, and eyes can be moved in different directions. And out of the 12 cranial nerves, it's interesting part, out of the 12 cranial nerves, 3 pairs of the cranial nerves are only for movement of the eyes. Eyes being so important structures, nature has given out of 12 pairs of cranial nerves, 3 pairs of cranial nerves are supplied to the eyes. I think we'll go for the eye of the goat now. So here I'm trying to use, this is the eye of the goat. This is the eye of the goat, and we have, of course, 2 eyes. Oh my God, the colors are looking different simply because we have punctured one of them purposefully. And this will be kept here. The eyes are placed in the socket like this. These are the sockets in which the eye is placed. And skulls of all these vertebrates, they have a socket in which the eye is located. And there's a lot of fat, behind the eye there is a lot of fat, as you can see here, as a cushioning provision. So that's how the eyes are put, and then there's a lot of fat. So I'll keep away this. I'll now deal only with the with this eye, and I think I can use this dish again for that purpose. And once again, you can expect this is this cornea part. Okay, and if you want me to expose the lens, I can take another eye here. And I hope I'll be able to help you to see the things deeper inside. Maybe here. I think it is better here. So I'll just give a cut here. and then yeah, I have given a cut and some pigment starts coming out. Can you see this dark pigment? It is actually of the choroid. The choroid seems to have been punctured, and so the black pigment. The choroid is the middle layer and it has got a lot of black pigment. It is because of this black pigment that light rays are restricted. I mean, otherwise there would have been reflections. You want refraction and image formation. You don't want reflection. I'll try to cut it now more strongly, and I think I'm going to show you the, oh my god, it's quite it's quite sturdy. Let us try to open it. Oh, I think a part of the lens is popping out. Some liquid. I'll be able to take out the lens now. That is damaged. I've not been able to pull out the lens correctly. I'm sorry for that. I got it messed up. So there is a lens inside. I think I'll be able to show the lens of the bird. So here, this is the eye of the bird, and here you can see once again the sclera, and then there's a black thing that is choroid. And then I think I'll be able to show you the lens here, which has already been dissected out, and I think it is left here. So this is the lens. Okay, this was here. I'll just put it here. You can see the lens here. This is the lens of the bird. It's seen to only a limited extent. And we can take a pause, and then with that, I think I got messed up with one of the eyes, but I think I'll be able to do this. And meantime, I have cut open the cornea, and I hope I will be able to go for searching of the lens. I'm sure it's going to be there. Okay, I'm able to do this now because already opened, and I'll just try to show you the lens now. That's quite interesting. I'll just hold this lens. Oh my god, oh yes, it is here. It is slipping off. Oh, I'm just trying to hold it, and oh my god, that's it. Oh, this is the great lens. Can you observe this? It's the lens of the bird eye. I'm sorry, not bird. This is a goat eye. So the lens of the goat eye. And I think I can also put it in the watch glass. I'll just put it here, and then I'll put it in the watch glass. This is a lens, biconvex lens. I can put it in the watch glass now. This dish, this circular dish, they are called as watch glass. So here you have the bird's lens, and this is the goat's lens. It's quite interesting. And human eyes will also have similar lens. And you know, you have heard of cataract, and that's a problem with the lens. And that's it. I'll just show you now the optic nerve. I think here I can show you the optic nerve, because optic nerve is starting from the eye, right? So here I can show you the optic nerve. I'll hold it. Can you see this optic nerve very clearly from the backside of the eye? This stalk-like structure. This one is the optic nerve. It is a sensory nerve. It starts from the eye and then it goes to the brain. It's a sensory nerve. All the fibers are sensory, and they will carry the electrical impulse to the brain. So optic nerve, since the second nerve, olfactory, optic, oculomotor. So this is the second nerve, optic nerve. That's it. Start. I think now we will try to understand something about color blindness. This is a very, very simple test known as Ishihara's test for color blindness. Color blindness, I think most of you know, it is a disorder which is known to be X-linked. There are two sex chromosomes, X and Y, and it seems that the X chromosome has certain forms of the genes which may be recessive in most cases, and those can generate some issues, some genetic disorders. And this is a kind of a genetic disorder, color blindness. What is color blindness? Color blindness is a situation where a person is not able to distinguish between red and green. It's very rare. It's very rare that a person is not able to see any color. That's not color blindness. Color blindness is usually restricted to red and green. I know they are not able to correctly identify red and green. Now it's very difficult for most of us to understand this, because we are able to understand what is red and what is green. But ultimately it is those cones, the different colors are possible due to different kinds of cones which are present, and their working genetically may become defective in some cases, and it is possible to relate those defects with a certain genes on the X chromosome. Without going into those details, I'll just tell you, we'll have a close look at these things. Please try to read this number. Or you would say, oh, it's so easy, it is 74. Yes, I'll read what is mentioned here. Ishihara's test for color blindness. In this test, the red-green colorblind person will read it as 21, and the normal person will read it as 74. I think most of you have been able to read this as 74 because you are not colorblind. And those who are colorblind, very interestingly, they will read it as 21. The spots are arranged in such a way that the test will automatically reveal whether the person is colorblind or not. I think this must have been a great job of the scientists who must have devised this. Like that, many such colorblindness tests are available. The percentage of the people with red-green colorblindness is very, very low. Very low. 1 in lakhs. Moreover, it's very interesting to know that colorblindness, if at all it is present in the population, it is almost always surely likely to be present in males, not in females. And there are reasons for this which we are going to learn. You have this topic for your 11th and 12th course later on, and we are going to talk about it. The only thing is that males have only one X chromosome because they are XY, and females have two X chromosomes, XX. So for certain reasons, females are less likely to be colorblind. Okay, same thing again. One more similar test. What number can you read here? I think you are able to read 42. Yeah. If you are reading this as 42, that means you don't have color blindness. In this test, the green colorblind person will read it as 4, and a red colorblind person will read it as 2. Oh my God. But we are all able to read it as 42. So that's color blindness. And I think in certain professions, they want people to be able to identify clearly the red and the green. And there are examples about these things and typical MCQs which will appear later on, how this is transmitted from parents to the next generation. But I think that is not our concern today. Last thing that I want to talk today is about a very simple test to know where is your blind spot. This is about your blind spot, This test is about blind spot. What is generally done is that, you can do this easily at home. Just take a paper and mark out such symbols. What you do is, see what I'm doing. Blind spot is something, when the optic nerve is making an exit from the eyeball, at this particular place, where the optic nerve is making an exit, rods and cones are absent. Rods and cones are absent where the optic nerve is making an exit. So there, if light rays fall, light rays can fall in different places. And if the light rays fall on this area, no image will be formed. So it is possible to know, and this is normal, everybody has that blind spot. What do you mean by blind spot? It is a concept where rods and cones are missing at the place, at the point where the optic nerve is making an exit, and that you can plot. This is a very simple model and many such models are easily available, and I very proudly mention that this is one simple model which has been given to us by our ophthalmologist friend Dr. Milind Dixit, and he has been a gold medalist from the BJ Medical College in ophthalmology, and he has been practicing in Nasik for several years. And yeah, his daughter is there with us in the standard 12, the current standard 12, and I think we have been very close friends, myself and Dr. Milind Dixit, and yes, he has been my student long back in 1993. Wonderful thing that I am enjoying my profession from that point of view, because I always feel nice to be linked with several people around. I will now tell you once coming to this blind spot concept. What you do is this way. Suppose I want to close one eye. Okay, I'll close one eye and I'll try to look at this plus sign. Okay, I'll try to concentrate on the plus sign. I'm closing one eye and I'm bringing this paper closer and closer slowly. Yeah, at this particular distance, I can't see the black spot. It means this time at this distance, the light rays are falling on my blind spot, and therefore this black spot is no more seen. I take it back again, I can see the black spot again. Now I can't see it. It is this way. You can just make a paper and try it. There are procedures available by which you can actually map the blind spot. But then I think that you can find out from net and you can do it for your purpose. I think with this much of work, it is good that we stop for the day.
विद्यार्थ्यांनी कॉमेंट्स लिहिलेले आहेत की ऍक्च्युअल ब्रेन पाहू शकतो का, तर बोट ब्रेन किंवा काही इतर ब्रेन, स्पीच ब्रेन वगैरे आपण नक्कीच पाहू शकतो. जर कुणाला पाहिजे असतील तर तुम्ही कॉमेंट बॉक्समध्ये तसं लिहा आणि ते आपल्याला दाखवण्याची नक्कीच व्यवस्था करता येईल. आपण सर्वांनी लेक्चर अटेंड केल्याबद्दल आपले सर्वांचे धन्यवाद. त्याचप्रमाणे आचार्य सरांनी आपल्याला डेमॉन्स्ट्रेशन आणि प्रॅक्टिकल सहित जे ब्रेन अॅनाटॉमी दाखवतील. त्याबद्दल सरांचे खूप खूप आभार, धन्यवाद. आता उद्याचा प्रोग्राम जो आहे तो फन विथ केमिस्ट्री, रविवार दिनांक 10 मे सकाळी 10 वाजता. आणि लेक्चर देणारे प्राध्यापक निलिमा उगावकर आणि प्राध्यापक संगीता रत्नपारखी. आणि त्या उद्याच्या लेक्चरची YouTube लिंक तुम्हाला लवकरच आपल्या ग्रुपवर मिळेल. धन्यवाद.